you on the right side of the wall? Do you see Phil Campbell? No, edit all, no.
I have no doubt that it's the future, right?
Just like, yeah. So the fact that they think they can make money off of it makes a sign that society There's a new Howard Hughes Local Trust Journal, SARS, all by one. There's PLOS One, which is the single largest publisher of private literature.
You know, for those I can say
yeah, I don't have any on that. Thank you. Some of those
well, because I recognize one or two.
some of them they say without asking. So I've written about this. Yeah, I've written about this in the book. One of these journals is big open access publisher.
That's one.
Yes. So they have on their editorial board, and then they made it up for children. They just copy. So they're just, pretending that just open access is just their little mantra. They're not.
But this is sending them.
Yeah, I mean, the problem is that some people have, just like the Nigerian email scam. People.
They do.
So we wanted to show you this actually. We wanted to create an open access journal of Nigerian financial scams. Yeah, exactly. I'm going to have to do that today.
Okay. Everybody, first thing. Is it?
I mean, it's
It's like not like up on YouTube.
yeah. It's not, and I think it's going to be good for you all. And it's like, there. The night before, despite the fact that we evacuated the next morning to go fire alarm, despite the fact that I spilled the head of his coffee on his suit right before my job, I was still Just relax.
All right. I think we can begin. I see many of you did not pay any attention to my recommendation last evening to stay home and miss our first speaker. I'm still just as jealous about his TED Talk, but I'm actually delighted to welcome Jonathan. He's professor in the Genome Center of Microbiology. I won't go on to all of his accolades. He has many, but I will say that were it not for an elbow that began to deteriorate in his pitching career at university, we would be watching him pitching for the Red Sox instead of being a microbiologist. So, fortunately, his elbow gave out before his microbiology did. So we're delighted to have Jonathan this morning.
It was my shoulder, by the way. All right, so thank you. Thanks for getting up early on a Saturday morning, especially after a wine festival yesterday. I'll try not to be too headache-inducing here. So I wasn't sure what the background and the style and the angle of everybody here is, because this is a conference on phenotyping, and I work on genotyping, which, you know, in many ways is obviously connected, although we frequently don't know how to make the connection between genotype and phenotype. But what I have been doing for 20 years or so is, how should I put this politely, sequencing the crap out of stuff, or sequencing crap, actually. I live and breathe DNA sequencing. That's really what I've been doing for 20 or so years. And we're very interested in function and phenotype. But every year when we think we're going to get into more functional studies, DNA sequencing gets cheaper and easier, and we keep expanding what we want to do with DNA sequencing as a precursor. So what I want to do is just try and give people a perspective on DNA sequencing. And I think you can use your imagination in part to think about the ways that this is going to be relevant to various aspects of phenotyping. And I'm going to give a little bit of background and then talk in the course of this about some of the things that people are doing with DNA sequencing. And because I was emailing back and forth with the organizers, they thought it would be useful to also talk about some of the new methods in DNA sequencing to give people a little bit of perspective of what's coming and why there's this sort of push to use sequencing in a lot of different ways. I'm going to talk a little bit about the sequencing methods. So if you look at DNA sequencing in studies of microbes or studies of various other organisms, but in particular microbes, you can really divide the progression into what you could call 4 eras of DNA sequencing. And it's not that they're in particular time order here, but they're somewhat in a time order, and they each continue after the other one started. So the first era, which in fact is still going on today, although it's being probably less used because it might be done, is the sequencing of one gene from microbes in order to place them into an evolutionary context. So this was started largely by Carl Woese in the 1970s, sequencing ribosomal RNA genes, which are highly conserved across all cellular organisms. And when you sequence these genes, you can compare the sequences to each other, calculate the similarity among the sequences, and build an evolutionary tree showing the relatedness among organisms. And this was and still is critically important, in particular for microbes, because phenotype of microbes is misleading as to their evolutionary relationships. And so for many years, as people tried to build a tree of life, evolutionary relationships among organisms, until the molecular biology revolution, people working on plants and animals focused on phenotype, some particular aspect of phenotype, and they could build reasonably accurate evolutionary trees of plants and animals and various groups. But for microbes in particular, bacteria and other microbes without nuclei, this was really hard. The phenotype just is not very informative, both because unrelated organisms become similar in their phenotype by convergent evolution, and because there weren't a lot of characters that you could use in the phenotype of many of these organisms that could then be used to infer phylogeny. And what Woese really showed was that if you use, in essence, as your phenotype, the DNA sequence, or the RNA sequence actually at the time, you could build evolutionary trees that were much more accurate. And this led to the discovery of a hidden group of organisms without nuclei, which he named the archaebacteria. They have subsequently been renamed the archaea. So this is the third branch in the tree of life, the third domain, as they're now called. And this was discovered entirely by building evolutionary trees from sequence data. And since that time, characterization of microorganisms in particular, as well as plants and animals and other multicellular organisms, have relied almost, now almost entirely upon molecular studies to infer evolutionary relationships. And ribosomal RNA is sort of the central theme of these molecular studies, but it's not the only gene that's being used. So the second era in studies of sequencing in microbes is taking this, what Woese had done, which was working on ribosomal RNA sequences from organisms you could grow in the lab, and expanding that to organisms you could study in the environment. So as I assume everyone here knows, many of the microbes on the planet can't be grown in pure culture in the laboratory, at least have not been grown in pure culture in the laboratory. And about 20 years ago or so now, Norm Pace in particular took the methods of Carl Woese of sequencing ribosomal RNA genes and went into environmental samples and started to sequence ribosomal RNA from those environmental samples. In fact, they started with sequencing RNA itself. And fortunately for them, there was an invention of the polymerase chain reaction, PCR, in the 1980s that came out. And although PCR was really designed for things like sequencing globin genes and sequencing HIV genes where you had, I assume most people know, but you have to have regions that flank the area in the genome that you are interested in, that you can make short oligonucleotide primers to amplify, make billions of copies of the region in between those two primers. And when it was originally designed, people were using this with, say, the globin gene from humans. And you could make conserved primers based upon known globin sequences. But what Pace and the other ribosomal RNA aficionados realized was that you could use conserved sequences in ribosomal RNA genes to amplify ribosomal RNA genes from unknown organisms at once. Any organism in your sample, you could potentially amplify the ribosomal RNA genes using PCR. And so they immediately went to environmental studies and environmental samples, collected a sample, extracted the DNA, did polymerase chain reaction amplification, and then you could sequence ribosomal RNA genes from organisms you could not grow. And then you can build an evolutionary tree of these and figure out what type of organisms are in your sample, even though you've never been able to grow them. And one of the things this revealed was that there were major lineages of organisms, entire phyla, that were not known in any of the culture collections. So phyla of bacteria and archaea and microbial eukaryotes that were only known from environmental samples. And probably a higher percentage of all of the branches in the tree of life were only known through these environmental samples and not through growing them in the laboratory. Another thing that this ribosomal RNA PCR was and still is very useful for is not just figuring out who is out there in a sample, but trying to characterize something about, for example, the relative abundance of those organisms. So you can sequence ribosomal RNA genes and then count how many times you see gene number 1 and gene number 2, and then you can estimate the abundance of the organisms that those genes came from the number of counts that you get in the ribosomal RNA sequences. And you can even do things like, this is a rarefaction curve where they're plotting number of what are called OTUs, a lovely jargon for the ribosomal RNA field. They're operational taxonomic units or species. You can plot how many you recover versus how many sequences you have read. And this can tell you something about how many organisms might be present in your sample. If you asymptote quickly, there are probably few organisms in your sample. If you don't asymptote quickly, there are a lot of organisms in your sample. And this, most people here are familiar with. I assume Chuck probably mentioned some things related to this. People can go into environmental samples and characterize the relative abundance of different taxa across large numbers of samples with ribosomal RNA PCR. Along the way, a few years after Pace started doing this, the next revolution really came by going back to the cultured organisms, the ones you could grow in the laboratory, and sequencing their genomes. And so in 1995, Tiger, the Institute for Genomic Research, which is where I worked for 8 years before moving to UC Davis, but before I got there, pioneered the use of this random shotgun sequencing to sequence the genomes of organisms. It had been applied previously by, in fact, Fred Sanger, who was one of the sequencing inventors, to PHX-174, a tiny little genome, where you take the DNA of an organism of interest, you shred it into lots of little pieces, and you read the sequences of random pieces of the genome of that organism. And for PHX-174, I think Sanger sat there looking at printouts of the sequence and tried to line them up with each other by hand. It wasn't that hard with 10,000 base pairs, although I wouldn't want to do it. And what Tiger did was merge, in essence, automated DNA sequencing, which I'll talk about in a few minutes, high throughput DNA sequencing with computational methods to do this for the whole genome of a free-living bacterium, Haemophilus influenza. So it was 1.2 million base pairs long. They shredded the DNA from this organism, randomly sequenced little bits of it, and lined up the overlapping pieces of the genome of this organism, and then did a little bit of gap filling until they assembled the complete genome of this organism. And this launched the genomics revolution. This random shotgun sequencing approach is now the routine method for sequencing the genome of any organism. This led to a big revolution in microbiology, so we've learned that what we thought we knew about microbes prior to genome sequencing was pretty narrow. One example of this is the extent of variation among closely related microbes. So this, you may or may not be able to see. Oh yeah, it does come out okay. This is a figure made from a comparison of the genomes of 3 strains of E. coli done by Nicole Perna and colleagues. So you take the gene content of these organisms, you compare them to each other, and they share about 3,000 genes with each other. And those genes that they share are nearly identical between these 3 representatives of E. coli. They line up in almost exactly the same order between the organisms, and they're 99+% identical at the DNA level. But what's amazing is that each of these strains has hundreds to thousands of genes not present in the other strains. So what is E. coli here is a little bit unusual. E. coli has a core that all the strains of E. coli share, but it also has a periphery that is variable among even closely related strains. Now, this is important for lots of reasons. One reason is that if you were using ribosomal RNA to characterize an environmental sample that contained E. coli, you would just say E. coli was there. You wouldn't know anything about the variation among these close relatives. They have basically identical ribosomal RNA genes. So massive genetic variation, even among closely related strains within a species, is one of the sort of fundamental insights provided by genome sequencing. And please stop me anywhere along the way here if there are any questions or if anything is unclear, if you want to know more. Yeah? What? Oh, so good question. Why did you chop them up? Because a couple of reasons. So Fred Blattner was trying to sequence the E. coli genome without chopping it up, and he was walking his way around the genome, sequencing one piece, and then reading the sequence and walking to the next piece. And that's how the public Human Genome Project was also working. The automated DNA sequencing technologies were so fast that what the rate-limiting step was feeding them with bits of DNA. And the easiest way to get lots and lots of bits of DNA to feed them was to just randomly chop up the genome of organisms and clone those pieces into E. coli and just feed those into the sequencing machines. That just accelerated the rate at which you could generate sequencing data. The randomness is really important when you stitch back together the genome, so you want your shredding of the genome to be as random as possible when you do this. But it was just mostly that the rate-limiting step was feeding the machines once the machines got faster. That make sense? All right, so another thing that, obviously for phenotyping and for people interested in phenotype, one great thing about genome data is you can do bioinformatics to make predictions about the phenotype of the organisms by annotating the genome and predicting the suite of metabolic and other functional properties within the genome of interest. Now, this doesn't work great. It works better and better every year. You obviously have to couple this to experimental studies. But for any organism, even if it's even if there's not a single paper on that organism, you can make a decent prediction of the suite of phenotypic properties of that organism from the genome sequence. So it is a nice first step in understanding the phenotype of a particular microbe, is making those predictions and guiding what you're going to do experimentally. So the fourth era in sequencing in microbes is the era that we're sort of in now, which is that the merging of the era 2 and era 3. So you go to an environment, you can't grow most of the organisms in the environment, so ribosomal RNA PCR was a way to figure out who was out there. You bring organisms back to the lab, genome sequencing is a way to make predictions about their function. So now we can integrate those two, and that's what's called metagenomics, where we go to an environmental sample and we just chop up the DNA and we sequence random bits of the DNA from the environmental sample and then hope or pray that we can sort through the data after we do this. It's a mess. It's very hard, but it is very, very powerful to do this. People have been applying this to all sorts of interesting environments. This is one of my favorite studies in early metagenomics by Ed DeLong, looking at a depth transect in the oceans. Ed DeLong was sort of the leader in this field, convinced a lot of people to do metagenomics because he basically used an environmental sequencing approach to discover a new class of phototrophy in the oceans, which sort of revolutionized ocean microbiology and catalyzed everybody trying to do metagenomic sequencing. So that's sort of the perspective on these different eras. And each of these eras still goes on today. We're still sequencing ribosomal RNA genes from cultured organisms to figure out what they are. We're still doing environmental PCR to figure out, with ribosomal RNA genes, to figure out who's out there. We're still doing genome sequencing, and now metagenomics is becoming hot. And what's sort of fundamentally important about each of these is sequencing costs and sequencing difficulty have gone down at a rate faster than Moore's law. So the cost of generating a base pair of DNA sequences plummeting, continues to plummet. And this makes DNA sequencing a technology that you can imagine a lot of other uses for. So for example, I mean, I assume most people are familiar with this, but if you're studying transcription in an organism of interest, whereas previously you might have done northerns or a microarray, now you can do RNA sequencing because the sequencing part is so cheap. And if you have a reference genome of the organism of interest, you can tile your RNA reads against that reference genome and use sequencing as a diagnostic of transcription, not of the genome of the organism of interest. And we can do this in every area. So if we go back now and look at those four eras that I outlined, we are going to sequence everything, by the way. Drew Endy at Stanford did a calculation as to when we will have been able to sequence every base pair on the planet. I think it's actually in only about 25 years from now. I argue with him about this because I wish him luck in finding every base pair on the planet. So in terms of sequencing the ribosomal RNAs of cultured organisms, if you can automate ribosomal RNA sequencing, the cost of doing this is so cheap that every single culture collection that anybody has in the laboratory, in the freezer or whatever, should have the ribosomal RNA genes sequenced from it in order to place those organisms in a richer context as to what they are. Much more important is this ribosomal RNA PCR from the environment. So for many years, there was a slow increase in ribosomal RNA sequence data in the sequence databases. You know, someone would go to an environment and sequence 100 ribosomal RNA genes, and that was a big study. So now with an Illumina HiSeq machine, many people, there was just a paper published where they did 3 billion ribosomal RNA sequences in an undergraduate project, right? So the amount of sequence data that we're generating is just completely ludicrous. And in terms of human-associated microbes, there is a revolution happening because of it. There's a European project called MetaHit. There's a US project called the Human Microbiome Project, and there are many other independent projects where we are building a map of the biogeography of microbes found in and on people in health and disease across time, across ages, across cultures, across different behavioral patterns. And ribosomal RNA sequencing is going to be a diagnostic. It doesn't necessarily say what is causing differences among people. But it is allowing you, associated with any study of human phenotype, to have the microbial list associated with that phenotype be easy to characterize. Even to the extent, I assume some people may have seen this, Rob Knight in Colorado, you could say this lucky or poor graduate student in his lab, had his face transected, dissected, whatever, where they did the biogeography of his face. And this was a couple of years ago. Now they're doing this for, I think, 2,000 people or something to that effect. So we are going to have a better and better roadmap of the microbes found in and on people, even differences between, say, the bridge of your nose and your ear. This is an ecosystem, a planet. Each person is basically a planet with, differences in humidity, differences in water level, differences in pH, differences in migration, differences in mucosal surfaces. Every aspect of our skin, of our internal systems, has a landscape. And the microbes that are going to be in there are going to vary across that landscape. So the same can be applied to genome sequencing. So as the sequencing technology got cheaper and cheaper, genome sequences are now becoming a routine aspect of studies of any particular microorganism. The genomes that have been sequenced are not being sequenced in the most systematic way. And for a few years now, I've been coordinating a project at the Department of Energy Joint Genome Institute to walk through the ribosomal RNA tree of life and fill in the gaps of genome sequences that are missing. So many of the genomes that have been sequenced were from a couple of the main lineages of bacteria, and we're filling in the rest, the things that people don't think are necessarily important, but for which their genome data is actually very useful. I'll come back to this in a minute, but the main limitation now in genome sequencing is acquiring the genomes of organisms that we haven't grown in the laboratory. There are only 6,000 described species of bacteria and archaea. They will all be sequenced in the next year, probably. It's the organisms that haven't been grown and described that we are having a hard time getting the genome sequence data from. And obviously, you can apply this to all sorts of areas, like the E. coli outbreak or various forensic studies. Genomics of microbes is being applied in all sorts of different areas. So metagenomics now is sort of the hot thing, although making sense out of the data is lagging far behind the sequencing of the data. But people are doing metagenomics now from every conceivable environment, just like they used to do ribosomal RNA sequencing from every conceivable environment. And in theory, we should be able to predict something about the organisms in a particular environment from the sequence data. I'm not going to go into the mess of analyzing metagenomic data. I'd be happy to talk to people about that. But right now, we're not doing a very, very good job of making sense out of the metagenomic data. It will come, but it's slow. The human ecosystem is being metagenomicized ad nauseum in good and bad ways. There was a really great study published about a year and a half ago now by the European group in collaboration with the Beijing Genome Institute with metagenomics of 120 fecal samples from 120 different people. They did all sorts of interesting things with the data. Most importantly is generating reference genomes for cultured and other uncultured organisms from an environmental sample. And once you generate those reference genomes, then the metagenomic analyses is much easier. So in the last couple of minutes, I just wanted to introduce you a little bit to what's going on in sequencing so you can have a feel for what you might use the sequencing technology to do. And again, creative thinking can apply sequencing to all sorts of new arenas because it is so cheap. So, the issues you might want to think about when you do this are cost per base pair, the read length. So the different sequencing technologies aren't all equal in terms of how long a piece of DNA that they're able to sequence. If you can get paired end pieces of DNA, so if you chop up the DNA from your organism into 10,000 base pair bits, if you read only one end of those 10,000 base pair bits, that's really the same as just reading a 100 base pair piece of DNA. But if you can read both ends and link them to each other, you now have a scaffold with which to do linkage mapping or haplotype mapping or genome assembly from your environmental sample. So I'm going to just go through quickly sort of the 4 eras, as I would call them, of sequencing as well. So the first era was really the manual sequencing. You could say Maxim Gilbert was also in this, but most people quickly avoided Maxim Gilbert sequencing and relied upon Sanger dideoxynucleotide sequencing, where you would manually pour and run a gel and then read the sequences in this ladder as they came off of the gel. What led to the genomics revolution was the automation of this by people like Lee Hood and a few companies, which changed the manual reading of the radiograms into fluorescent automated reading of capillaries with your DNA sequence data. And you were able to make this very fast because of this fluorescent automated reading of the sequences coming off of these machines. An important aspect, this goes to your question, as the fluorescent automated sequencing machines started to come out, it became very hard to keep up with them, to feed them. And so another aspect of automation was automation of cloning the DNA. So the way this worked was you would take DNA from an organism of interest, clone it into E. coli usually, or sometimes other organisms, grow up each of those individual clones in, say, microtiter dishes, extract DNA from each, the plasmids from each of those, and then sequence those. So one part that was rate limiting was picking colonies. So we used to have what we called massively parallel undergraduates doing colony picking, and they were not very happy with this. And very rapidly people developed or took advantage of existing automated colony picking robots that would take a plate and rapidly take a picture of it and then pick individual colonies. And once we were able to do that, the cost for an individual sequencing read was about $1 for 1,000 base pairs of sequence. And again, doing it in a systematic way where you walked your way around the genome, you could only generate enough clones to feed, maybe 100 sequencing reactions. At Tiger, where I worked, we had 100 ABI 3700 sequencing machines. The rate limiting step was feeding them, was getting clones to feed those machines. And automated Sanger sequencing is what led to a lot of the revolution in genomics, in ribosomal RNA studies, and in metagenomics. And what is now going on is this, what you could call the 3rd generation of sequencing, which there are many different approaches to this, but the key fundamental point is we no longer clone individual pieces of DNA. The methods that we use are able to generate sequence data from thousands to millions of individual pieces of DNA without ever having to go through the laborious step of cloning those pieces into E. coli and growing the E. coli. So there are 3 main methods that people have been using for this. Illumina used to be called Solexa. Illumina bought the company Solexa. Roche bought the company 454. And Applied Biosystems developed their own methods. So this is Illumina sequencing, 454 sequencing, and ABI solid sequencing. And I could go into all the details of how they work. There's this great review paper by Elaine Martis, if you want to look into it more. She's the head of the Washington University Genome Sequencing Facility, and she details exactly how the methods work in all of these methods. Very well written, very clear. She also has a YouTube video online where she's describing all of these. It's really great. And I'm not going to go into all the details of how they work, but basically they differ in how they make. So what they do is you shred up the DNA. You usually do some library construction modification to add some special little pieces of DNA for subsequent sequencing steps. And then you need to isolate a piece of DNA and make millions of copies of that piece of DNA in isolation to then run the sequencing reaction. So for example, 454 sequencing encapsulates little pieces of DNA in these emulsion beads. And then you run PCR in those emulsion beads to make lots of copies of each individual piece that's inside a bead. And then you place those beads into a little grid, and you run a sequencing reaction where you're reading the sequence of the millions of copies of each of those beads. Illumina does this in a different way. It layers the DNA onto a slide, but the principle is the same. to take a single piece of DNA and then make millions of or billions of copies of it that allows you to enhance the signal that you would get when you then run a sequencing reaction on those little pieces. So I'm going to show you a picture. I'm going to skip through this and, oh, I must have taken out the pictures. The details don't really matter that much, but in essence, what these methods are doing is tricks to try and isolate your single pieces and make lots of copies of it. In my opinion, Illumina has won the game here. If you compare the methods to each other, you can compare their chemistry, how they work, how they do this amplification, how long the sequences they're able to get off of this, the accuracy of the sequences, etc. And each of the methods has their use, but Illumina has gone kind of crazy in terms of the number of sequences you're able to get in a single run. So in the HiSeq, the new Illumina HiSeq machines, you're able to generate billions of sequences in a run of a machine. And they even have, each of these companies are making mini versions of their machines. The Illumina MySeq machine, which is their mini version of the machine, has more capacity for sequencing than the entire operation we had at Tiger 5 years ago. One little $150,000 machine. So, you can still look into how to use these, but for example, for Illumina, for $10,000, you can generate billions of sequences. So the cost per sequence is vanishingly small compared to the cost of doing other things in a project. And what I'll just end with two things. For taking advantage of the DNA sequencing revolution, just as it was with the automated fluorescence sequencing, the key now is really sample preparation. How can you feed the machines and take advantage of their capacity? So for example, for Illumina sequencing, what we want to do is take hundreds to thousands to millions of samples, and in each sample, add a little barcode to the DNA in that sample. A little DNA sequence barcode, say AACC, AACC for sample 1, and AAGG, AAGG for sample 2. We add that to our DNA, we mix it all together, and we sequence it, and then bioinformatically we figure out which sequence came from which sample based upon the barcode. And we can combine together pairs of barcodes to generate millions of different sample tags. And that's how we're going to keep up with these machines, because for many studies, you don't really need more than 1,000 or 10,000 sequences to characterize a sample. What you need is to do hundreds of thousands of samples across the human body, different time points, different patients, etc. And figuring out ways to feed the machines and take advantage of their capacity but do millions of samples is currently the revolution that is going on. And there are many companies that are selling kits to help you do this. There are many tricks for doing this. Barcoding. And what I just want to end with is what I would call generation 3.5 or generation 4. There are other methods out there you may have heard of. Generation 3.5 is this amazing ion torrent system, which still has some challenges in terms of its accuracy, but it is incredibly fast once you feed DNA into the machine. And it's reading sequence instead of by fluorescence, but by a computer chip, by changes in electrical charge that are generated when you remove a hydrogen group, for example, from an A as opposed to from a C. Absolutely amazing technology, although the accuracy is still challenged. And then generation 4, which is still a work in progress, is single molecule sequencing. So instead of having to make millions or billions of copies of each individual piece of DNA, wouldn't it be great if you could just read the sequence of individual pieces of DNA? And that's what the new technology is trying to do. There's a company called Pacific Biosciences. We have one of these machines in the Genome Center at UC Davis, where you read, you watch in real time as a DNA polymerase copies a single molecule of DNA. And because you're watching in DNA polymerase time, it's incredibly fast. Instead of 15 bases a day, which is what Illumina, the HiSeq machine, does, this is 15 bases a second. And Pacific Biosciences is generating reads of over 10,000 base pairs in some cases. In theory, it can do 100,000 base pair reads on a single piece of DNA. The accuracy compares very well to rolling dice, not very well to any other sequencing method. The accuracy is about 87% or something. It's really poor right now. If you, however, generate data with Illumina as your backbone data, and you use specific biosciences to just build a scaffold, you can figure out where you are in the Illumina data from bad quality sequence, and then you can figure out the larger ordering of the genome from the PacBio data. PacBio is also very interesting in that because it's watching in real time, it has the potential to detect things like methylation status, because that slows down the DNA polymerase. So many people are using PacBio for things like epigenetics to look at methylation status, and we'd like to use it to look at DNA damage state in pieces of DNA. So it has a lot of potential, but the accuracy is still a nightmare challenge. And then the last one I just want to mention, which you may have heard of, is this nanopore sequencing, which is just freaking ridiculous. So this has been around for a while. There's a company in England that's been trying to do this, and a few other places that have been trying to do this. In essence, the idea is to take a membrane channel, pull DNA through the membrane channel, and detect the sequence by some sort of alteration in either the membrane channel protein or in some conductivity in that protein or in something you can bind to those proteins that are the membrane channel. An Oxford Nanopore company announced a few months ago that they actually had gotten this to work. And more amazingly, what they announced was that they can do this in a thumb drive, a disposable thumb drive sequencer, where you literally just sprinkle your lysed cell into the hole on the thumb drive, and it reads the sequence and puts it onto your computer. They haven't sold any of these yet. They haven't released these to anybody out there, but in theory, they may actually be able to do this, and that will lead to another revolution, which is field DNA sequencing or clinical sequencing with, you don't have to buy a $200,000 machine, you buy a $700 thumb drive. So I think I'll end there. There is this revolution going on. That's what I want to convince you of. Sequencing is so cheap that you can use it as a diagnostic for characterizing all sorts of aspects of organisms. And again, not just their DNA, but their RNA too. And many people are manipulating DNA sequencing to study things like protein, transcription factor binding to various regions of the genome. And if you integrate this with other technologies such as metabolomics, proteomics, high-throughput genetics, high-throughput phenotyping, etc., then we're really going to have a full sort of comprehensive understanding of the microbes that are in any particular environment and that have been recalcitrant to a lot of studies because the phenotype has been hard or misleading and the genotype has been invisible. So I think I'll just end it there. Thanks.